Now we will change the assay from RNA to ATAC because we have done the cell type annotation, and then we transferred these annotations from single cell RNA-seq data to single cell ATAC-seq data. So we don't need RNA data anymore. So we will also change the active assay from RNA to ATAC, because we will use the ATAC for downstream analysis. So right now you can see the default assay. We will use the default assay function to check the assay. So PBMC and run. You can see our active assay is RNA, but now we will change these from RNA to ATAC because we don't need RNA anymore. So default assay and run. There is an error in the code, so we will look into the available assays, and you can see only two assays are available. One is the RNA assay and the another is the PICS. So there is no assay with the name of ATAC. So we will change the code to from ATAC to PICS because that is our real ATAC assay. So PICS and run. Now we will check again that whether the default assay has been changed from the RNA to PICS or not. So we will use again the default assay function. So default assay PBMC and run. Now you can see the default assay from RNA to PICS has been changed. From RNA to PICS has been changed. Now we will find the differential expression of PICS between two cell types or groups. So differential expression of PICS between two cell types or groups. So we will use the given line of code. So DA PICS, and we will use the find marker function from Seurat object to compare two cell types or groups. So we will use the two types of cells. You can type any types of cells. So I will type CD4 cells as in ident1, and the CD4 plus monocytes, which is the reference group, and you can use any two cell types or groups. We will use the Wilcox test by default. main.pct equal to 1, and the minimum PCT 0.1 is the threshold here, which means that the test peaks that are detected and at least 10% of cells in one group compared to another group. So we will use this line of code, and this line of code will find the differentially accessible peaks between the genomic regions that are significantly more or less open in one cell type compared to another cell type. So now we will press the run button. We got some errors because the find all markers function we will use the find markers function, and also we need to type the Seurat object that the find markers for function from the Seurat object need. So pbmc, and now we will run the code, and hopefully this time the code will run. Error again. Now the code is this time successfully running. It will take some time, and this line of code will find the differentially accessible peaks genomic regions that are significantly more or less open in one cell type compared to another cell type. And we will use the find markers function from Seurat package for this task. So it will take some time. Now you can see the code has completed running, and it took a somewhat long time of 37 minutes. So now we will check the value of differential peaks for few rows that are significantly different, and the two groups are cell types. So we will use the head function. So head peaks and run. Now you can see the values. So for first few rows, and the p-value is the raw p-value from the Wilcoxon rank test, and similarly average log to fold change value is also given, and the positive value of average log to fold change means a higher accessibility in ident1 or cell type 1, which is CD4 plus cells, as compared to other cell type which is monocytes. And if its value is negative, it well means a higher accessibility of peaks in ident2, which is monocytes, as compared to ident1, which is CD4 plus cells. Similarly, the PCT1 is the percentage of cells in ident1 with non-zero accessibility, and the PCT2 is the percentage of ident2 are cells type 2 or monocytes with non-zero accessibility. After performing the differential expression analysis of peaks, now we will generate some beautiful plots. So I'm using the given line of code for a Weiland plot and a feature plot. So I I am generating a plots for the row first, which is the first picks. So you can generate the plots for any picks. For example, you can type 2, you can type here 3 and 4. So now we will generate some beautiful plots to compare the two cell types or groups for differential peaks analysis. So now first of all I will run this code for a while and plot, and now I will generate a feature plot. So for a sidewise visualization, I will and plot 2 and run. Now you can see, let's zoom in. So you can see the Wyland plot and the feature plot has been generated for both the cell types or groups for pix 1. So you can also generate such type of plots, and you can interpret the plots as according to your research hypothesis and plots. So now we will show some peaks because there would be a lot of peaks and we cannot show all the peaks, so because it would be time consuming. So we will show some peaks that really matters between the two groups, between the two cell types. So we will use the given line of code. So we will use the given line of code to show peaks that really matters between the T cell and monocytes. Here the log 2 FC3 means that peaks in one group should be 8-fold higher accessible than another group. So now we will run the code one by one. So it will return some important peaks that really matter and will save your time. So and now we will show the peaks that really matter for the T cell. So we will show few rows of peaks. So these will return also the name of the peaks. So run. You can see these are the peaks that really matters for the T cells, and these are 8-fold higher accessible and T cells than and the CD4 monocytes. So now we will show the peaks for now for the monocytes. So and these you can see the output in the console, and these are the peaks that really matters for the monocytes. And these peaks, differential peaks, would have higher accessibility and the monocytes compared to the T cell. So you can also check and see the name of those peaks. So I have showed you the differential accessible peaks or genomic regions between the two groups or cell types, And I have also showed you the genes that are existing and those differential accessible peaks.